Hello, everyone. My name is Hunter Prude, and today I'll be presenting some of the research that I worked on for my master's thesis at New Mexico State University, where I examined the influence that man-made water sources have on puma kill site locations and prey composition. So to get started, since the mid-1900s, state and federal agencies have developed man-made water sources for wildlife in arid regions. In 1999, Rosenstock reported that there are more than 6,000 man-made wildlife water sources in the Southwest that cost more than $1 million annually to maintain. Although wildlife are generally considered to benefit from man-made water development, there's very little known about how man-made water sources impact predator-prey dynamics. So there are two primary hypotheses through which man-made water sources might influence predation. First is a prey abundance hypothesis. This is where predators will select for areas that have increased densities of prey. So man-made water sources tend to aggregate prey species during the drier seasons. Therefore, it is possible that water sources can become predation sinks due to the increased abundance of prey during those dry seasons. Next, we have the prey vulnerability hypothesis. This is where predators will select for areas that have increased stalking or ambush cover. So it's possible that the habitat features surrounding man-made water developments might actually increase predation risk for wildlife. So to examine this, I compiled puma kill data from 7 southwestern study areas that varied in temperature, precipitation, aridity, and availability of man-made water sources. My objectives were to determine the relationships between the locations of puma kills with the proximity of man-made water sources, and then to build upon that relationship by evaluating the influence that other environmental and topographical factors have on the timing and location of puma kills. So these two images on the right are a good example of what I was trying to evaluate. So in this top image, you see a mule deer buck getting a drink of water at a man-made water source. 30 seconds later in the bottom picture, you see a puma stalking that buck from that water source. For the study areas, if you look at this map on the right, each one of those polygons represents the 90% kernel home range of all the combined puma data within each study area. So we had 3 areas in the Chihuahuan Desert, New Mexico. Those included the Armendaris Ranch, the Ladder Ranch, and then the Sevieta National Wildlife Refuge. So these 3 areas are hot and dry, but they do have some natural water sources available for wildlife use. So next we had 2 areas in the Sonoran Desert in Arizona. Those included the Bighorn Vulture Mountains area and also the Kofa National Wildlife Refuge. So these 2 areas are extremely hot and extremely dry, and they have very few, if any, natural water sources available for wildlife use that haven't been modified by a man-made structure. Then we had 2 non-desert areas in New Mexico. Those included the Guayanus Mountains area and then the Jemez Mountains area. On average, these 2 areas receive more annual precipitation. They have cooler temperatures and then more available natural water sources than the desert study areas. For my data collection, pumas were captured using traps or hounds and collared with GPS Iridium satellite collars from 2007 to 2018. Diet composition data was collected for each puma through the field investigation and GPS clusters. In general, clusters were just defined as any location in which a puma spent 6 or more hours within a 100-meter radius. I obtained the locations of water sources from maps, historical records, satellite imagery, ArcGIS data, and sometimes field observations. Most of these study areas kept updated maps or GIS files of water developments. However, the private lands surrounding the study areas often had few records if they had any records at all. So for these areas, I would use a handheld GPS unit to mark water locations while I investigated clusters in those areas, or I would use satellite imagery to remote sense any water sources for the areas that I did not have access to. For my analysis, the primary purpose of this study was to determine if pumas were using man-made water sources to ambush managed game species, primarily ungulates. Therefore, I only included data from ungulate kills in the analysis. So the ungulate species that I included were deer, Barbary sheep, bighorn sheep, elk, feral burro, feral horse, javelina, oryx, and pronghorn. So to determine if the spatial distribution of puma kill sites were associated with the locations of perennial water sources or other environmental factors, I created random points for comparison. So I used ArcGIS to calculate the 90% kernel home range for each individual puma, and then I created randomly distributed points within that puma's home range using a ratio of 20 random points for each kill documented in that puma's data. I then replicated the data derived from the documented puma prey kills 20 times and then randomly assigned it to those randomly generated points. So pumas often drag their prey from the point of ambush or kill site to the nearest concealment cover before they feed on the carcass. And because of this, the actual kill location, or where the prey animal was attacked and killed, is often difficult to find in the field. And so many times you just record the carcass location in the data. So to account for the variability and habitat characteristics between the kill and carcass locations, I use ArcGIS to create 100-meter buffers around all of the kill and random point locations. I then use those buffer areas to extract the habitat characteristics at kill sites and the random points. Prior to the analysis, I assessed the data for multicollinearity using Spearman's rank correlation coefficients. When two variables were highly correlated, I would remove one of those from the analysis. All of the remaining continuous covariates were then standardized. The covariates that I included in the analysis were Palmer Drought Severity Index, average temperature, woody cover, visibility, distance to man-made water sources, terrain ruggedness, season, study area, estimated vegetation type, and prey species. I then subset the data into 7 different data sets. Those included the combined study areas, the Sonoran desert areas, the Chihuahuan desert areas, combined desert areas, non-desert areas, only deer kills, and then only bighorn sheep kills at areas that had populations of bighorn sheep. I developed a set of a priori model structures that included distance to man-made water sources and then other habitat characteristics that could contribute to predation risk for prey species or also contribute to stalking cover for pumas. So the models that I developed are all listed in this table to the right. I then used mixed effects logistic regression to determine if the probability of a site being a kill location was related to the proximity to water sources or other habitat characteristics. So because determining if man-made water sources influence puma kill site locations was one of my primary objectives, I included distance to man-made water sources in most of the models. That said, woody cover ranked pretty highly in most of the preliminary model results. So I also developed some models that included woody cover without distance to man-made water sources to determine if woody cover had a greater influence on the probability of a site being killed rather than water sources. I used a Kiki's information criterion corrected for small sample size to rank the models and then to assess model support. I considered any models with a delta AIC from 2 or less to be competing model, but also considered any models with an AIC from 2 to 7 to have some support. I calculated variance inflation factors for the covariates in the top ranking models and considered any models with values exceeding 5 to be correlated. I then evaluated the predictive performance of each of the highest ranking models using K-fold cross-validation. So for my results, diet and spatial data was collected from 82 collared pumas from 2007 to 2018. There were 1,556 documented ungulate kills and then 31,120 random locations used for this analysis. The majority of the kills were mule deer followed by elk and then bighorn sheep. Kills occurred closer to man-made water sources during the hot and dry seasons at most of the study areas, the exceptions to that being the Jemez Mountains and the Ladder Ranch, which both of those study areas actually have increased ephemeral water during the hot, dry season due to snow melt runoff. So of the 1,556 ungulate kills documented, only 20 of those kills actually occurred within 100 meters of a water source and might have been instances of a puma using a water source to ambush prey. The majority of the kills occurred between 1 and 2 kilometers of a water source, and then 86% of the total kills occurred within 5 kilometers of a water source. Prey kills occurred at closer proximities to man-made water sources than the random locations at all of the desert study areas. The random sites were actually closer at the 2 non-desert areas. The mean distance between kills and water was greater than 2 kilometers at all the study areas, except for the Ladder Ranch, where the mean proximity was 1.1 kilometers. Javelina, pronghorn, and elk kills occurred closest to water sources. Bighorn sheep, mule deer, and oryx kills were located at medium distances from water sources. And then Barbary sheep, feral horse, and feral burro were located furthest from water sources. So for the statistical analysis results, for the all study areas data set, the highest ranking model included distance to man-made water sources, woody cover, and drought severity, and then a significant 3-way interaction between those 3 covariates. The K-fold predicted performance estimation for this model was very good. So at all the study areas, the probability of a site being a kill generally decreased with increasing distance to man-made water sources and then increased with increasing woody cover. So at the lowest levels of woody cover, the probability that a site was a kill decreased with increasing distance to man-made water sources for all levels of drought severity. As woody cover increased, the probability that a site was a kill increased with increasing distance to man-made water sources during periods of drought severity near and above the long-term mean. So during drier periods, the probability that a site was a kill decreased with increasing distance to man-made water sources, regardless of the changes in woody cover. During wetter periods, the probability that a site was a kill increased with increasing distance to man-made water sources. There were 3 competitive models for ungulate kills in the Sonoran Desert. The variables in the competitive models included distance to water, woody cover, and average temperature. The top ranking model had 42 to 75% more support than the other 2 top models, and the predictive performance estimation for these 3 models was good. So based on the parameter estimates for these 3 models, they indicated that the probability of a site being a kill in the Sonoran Desert decreased with increasing distance to water and then increased with increasing woody cover. This was especially true during drier periods. The best performing model in the Chihuahuan Desert study areas had overwhelming support and included distance to water, visibility, and then had an interaction term between distance to water and visibility. The predictive performance for this model was also very good. So in the Chihuahuan Desert, the probability of a site being a kill decreased with increasing distance to water sources and then decreased with increasing visibility. The effect of distance to water generally increased with decreasing visibility. There were 2 competitive models for the non-desert study areas. Both of these models included distance to water and woody cover, while the second ranking model also included drought severity. The highest ranking model had twice as much support as the second highest ranking model. So in the non-desert study areas, the probability of a site being a kill decreased with increasing distance to water and then increased with increasing woody cover. The influence to distance to water became more pronounced with increasing woody cover. The proximity to water sources increased in areas that had more woody cover, especially during drier periods. The results for deer kill sites were very similar to the results for the all study areas data set. These results indicated that the probability of a location being a deer kill was related to distance to water, woody cover, and drought severity. So at the lowest levels of woody cover, the probability of a site being a deer kill decreased with increasing distance to water sources for all levels of drought severity. As woody cover increased, the probability that a site was a kill increased with increasing distance to water sources during periods of normal and high precipitation. During dry periods, the probability that a site was a kill decreased with increasing distance to water sources, regardless of the changes in woody cover. There was high model selection uncertainty and then 4 competitive models for bighorn sheep kills. The top ranking model included distance to water without an interaction term. The second ranking model included an interaction term between distance to water and season. The third ranking model had an interaction term between distance to water, average temperature, and woody cover. And then the fourth ranking model had an interaction term between distance to water and woody cover. So for bighorn sheep, the probability of a site being a kill increased at closer proximities to water in areas that had more woody cover and varied across seasons. The probability of a site being a kill near water sources was highest during the hot, dry season. But for all seasons, the probability of a site being a kill decreased with increased distance from water sources. So these results indicate that the locations of puma kills are actually influenced by proximity to man-made water sources and also habitat characteristics, primarily ambush cover. So this provides supportive evidence for both the prey vulnerability and the prey abundance hypothesis. The lack of kills within 500 meters of a water source suggests that there is not a significant amount of puma predation on ungulates at water sources. Therefore, pumas do not appear to be using man-made water sources as hunting locations. However, the mean proximities of puma kills to man-made water were closer than the random locations within the desert study areas. So this indicates that man-made water sources do influence where puma encounter and kill prey. I did find consistent relationships suggesting that the probability of a kill declines with increasing distance to man-made water sources. So this suggests that the primary mechanism driving the influence of water sources on the locations of puma kills is likely through the influence of water sources on the distribution of prey on the landscape. not necessarily that pumas are using water sources as a means to locate and kill prey. The mean proximities to kills and water were consistent with the mean proximities of collared animals to water documented in some previous studies. So Krausman had reported that the mean distance of mule deer locations to water catchments range from 1.9 to 3.3 kilometers in the Bighorn Vulture Mountains area, where we found the mean proximity of mule deer kills being 2.8 kilometers from water sources within that area, and then 2.9 kilometers at all the combined study areas. Kane reported that the mean proximity of bighorn sheep locations to water catchments ranged from 2.5 to 2.9 kilometers in southwestern Arizona, which is also consistent with our findings of the mean proximity of bighorn kills to water being 2.5 kilometers at all the study areas. So many previous studies have identified stalking cover to be a critical component for pumas to be able to successfully capture prey, and most of these studies have documented pumas using woody vegetation as their primary source of stalking cover. So the results from this study provide additional evidence that pumas select for areas that have increased stalking cover provided by woody vegetation to ambush prey, because woody cover was in many of our highest ranking models. However, the models that had woody cover without distance to water did not perform well. So this indicates that woody cover alone does not explain the distribution of puma kills on the landscape. Drought severity, or average temperature, and distance to water were also included in most of the high-ranking models. So this indicates that aridity strongly affected the water requirements of prey and influenced their distribution on the landscape by restricting them to areas near man-made water sources. It was expected that drought severity and temperature would influence kill proximity to water in the desert study areas. However, drought severity was also a predictor for kill proximity to water in the non-desert areas that generally have cooler temperatures, receive more precipitation, and then have more available natural water sources. So this indicates that prey are still dependent upon man-made water sources during the dry and hot seasons, even in areas that have increased availability of natural water sources. To summarize all of this, in the arid regions of the southwestern United States, pumas do not appear to be exploiting the predictable prey visitation of man-made water sources, but they are capitalizing on the increased densities of prey in the vicinities of water sources. So on a landscape scale, pumas will select for areas proximate to water sources that have increased prey abundance and higher probabilities of encountering prey for hunting. Within that landscape, pumas will select for fine-scale habitat features such as woody cover that enhance their ability to stalk, ambush, and effectively kill prey. Climatic conditions such as drought severity and average temperature will influence the distribution of prey on the landscape by restricting them to areas closer to man-made water sources during the drier and hotter periods. Therefore, puma kill site locations will likely occur at closer proximities to man-made water sources during those dry and hot seasons. This is a list of the literature cited and some acknowledgements. And with that, if I have time, I'll take any questions.